concerned, we have nucleic acids and proteins which are going to play a critical role in our biological system. So protein sequences are extremely important, and we do have primary structure, we have secondary structure, tertiary structure, and then quaternary structure. Usually what we do, we believe that whenever we have a sequence of a protein molecule, it is ultimately going to result in production of a structure, and that structure is critical for function. So various different tools, open source tools, which we use in our research and for our teaching at our bioinformatics classes, they include different types of databases. I'm pretty sure those of you who are familiar with biological sciences, they know that there is a main resource of NCBI which has various different databases available. And we can use them in our studies, and these databases are used for depositing our data. We can retrieve our information, and then we can also analyze them. When we try to talk about sequence comparisons, the area that we do. In case of sequence alignment, we can use different open source software. Cluster Omega is one of them, and here you can see the screenshot of Cluster Omega. And what we do, we submit our sequence, and then we are going to get overall alignment. So basically, we are interested in comparing our sequence with other sequences in order to understand what is happening in our protein sequence. Last is another very important tool that can be utilized, and we are interested in comparing our sequence of interest with that of other sequences which are present in database. So again, you submit your info, and then you're going to get overall results. In case of phylogenetic analysis, if we are interested in comparing our sequences with various other sequences present during process of evolution, we can use again various different phylogenetic software. Phylet is one of the software which is open source and freely available. And using this software is going to give us information regarding what overall distance between different organisms, and we can get info about our homologous sequences and analysis. One of the very important area is motif search, and in case of motif search, we can use PFAM. Again, we can provide our sequence for searching, and what it does, this particular database, a search engine is going to compare your sequence of interest with that of other sequences, and ultimately what we can do, we can get information about already available information. We can get information about that. Three-dimensional structure of protein is extremely important and critical because that is going to give us information about overall functionalities, and mostly proteins are involved in various types of disorders and diseases, so we are interested in knowing the structure so that we can develop various drugs against them. So there are again various open source tools which are available for secondary structure prediction, in which we usually use programs like SciPrep and all, and you can get information about overall secondary structure, that is overall helical structures or beta-pleated sheets and all. As far as tertiary structures are concerned, they are very important again, and one of the very famous software which is available for free for academic users is Modula. And what we try to do, we try to perform homology modeling in which we use our sequence of interest, and then we try to utilize a specific template with the help of which we can construct a model, and then that model can be used for further analysis. As you can see in this picture, that the overall structure of protein has been predicted using computational tools, and these are again free softwares which are available. What we can do using these software is you can perform superposition also. That is, if you have one molecule available, you superpose it with using another molecule and you can compare overall structure. So ideally, if you are doing something using computational tool and something that has been done experimentally, you can compare and analyze that how closely associated they are. So these types of superposition tools are extremely critical because they are going to provide you information about how good or how bad your softwares are working. So ultimately, computation models and experimental models can be compared by superposing. And here is another figure which simply indicates that superposition of the model which was constructed using computational program and then experimental, how well they are going to superpose on each other. Another very important area in structure analysis is drug designing and docking. As all of us know that various diseases are kind of like affecting all human beings, and we are very much interested in designing various drugs, and interestingly most of the proteins are involved in these types of disorders. So using computational tools, one can perform drug designing and docking. So in this case, what we do, we have either a small drug molecule or a small organic substance which is docked or attached on top of a receptor, which is usually a protein molecule. So one can perform receptor ligand drug designing, and this is a screenshot of the program which is freely available, which we very much use in our research. This is Autodoc tools. What we do, we use a particular structure, three-dimensional structure of a protein molecule or receptor, and then we try to dock molecule of our drug onto it. This slide shows you overall docking patterns of 4 different drugs. This is the work which we conducted in case of oral cancer. You know, oral cancer is very much common in Pakistan, and particularly our younger generation, they are more affected. So, and because it is kind of like diagnosed at later stages, so it's not easy to treat them as well. So we were interested in identifying comparatively new drugs for it. So what we did, we used epidermal growth factor receptor structure, and we tried to dock various drug molecules onto it, and we examined overall energy scores and all, and by comparing these, we identified that varitinib was the best drug which was giving us good scores. So we went into the lab and then we performed further analysis, and we got very interesting results. But because this forum is not for sharing those results, so I'm not discussing our results here. However, what I want to highlight is that we can utilize open source softwares in order to explain and understand what is happening at the receptor drug level. PairDoc is another software which can be utilized, and here you can see this is the work which we conducted on HCV, and we tried to develop various drugs using computational tools against, using fluoroquinolones and natural derivatives, and we used PairDoc software in order to analyze this, and then we got very interesting results. And then after identification of various natural rocks, we went to the lab and tried to test them out. Proteins do not work in alone. They are interested in working in association with other proteins. So protein-protein interaction softwares are also available. String is one of the very common software which is capable of giving us information. So what we do in string software, we try to base all our proteins and genes names, and then what you can do, you can get information about their overall interaction, that how closely associated these different proteins are with each other, or how far they are, and what is exactly their overall classification tool. So you can perform these types of studies as well. There are various online portals and software available which can be utilized by different biologists and biochemists. For example, one of the very common one is Expressi, and this Expressi software has its portal has various different options available, and you can click on different things on your extreme left hand side. You can see there are various options available. You can click and you would see different possibilities available. So in proteins and proteomics, there are various resources and different programs which can be utilized, and our students commonly use all of these resources. Similarly, we use a program which is BioEdit, and this provides a variety of different opportunities and options for our students, so one can analyze and identify various aspects of different macromolecules, whether they are protein or they are nucleic acids. There are different visualizing softwares available. For example, GelView, which is going to give you beautiful graphics. You can perform three-dimensional analysis, or you can check overall comparatives at multiple sequence alignments and all, or distance matters. Similarly, there is another very good software which is Discovery Studio Visualizer. And what you can do, you can visualize a three-dimensional structure protein molecule into it, and this would allow you to zoom in, translate, rotate, and everything. So you can identify, you can look at different types of structures and analyze and identify what is happening at molecular level. So overall, take-home message for biologists is that there are multiple open source online resources available, and one can use them to infer information about our molecule of interest, and ultimately this information is obviously going to be very important when we are going to do it or analyze computational tools along with our wet lab experiments. So this is going to help us in understanding more about overall molecules. I thank you very much for your attention.